to give you a little bit of background about me, why it is I get to stand up here and talk to people about life in the soil. I have spent the last 40 years working on soil biology. Starting out at, when I was an undergraduate student, we didn't know about life in the soil. We had no idea why protozoa, what the good guy nematodes, we didn't even call them good guy nematodes back then. Why are there fungal-feeding nematodes? Why are there predatory nematodes? Why are there bacterial-feeding nematodes? Why do we have flagellates and amoebae and ciliates? Who really cares? Microarthropods, of what importance could those possibly be? Yeah, just try growing plants without those organisms in your soil. But it took a long time to develop this knowledge and understanding, to understand what the role and functions of all these organisms are in the soil. So I want to briefly describe that. What is the soil food web? A little bit of information about direct microscopy, because here's the simple, easy way for you to be assessing your own organisms in your own soil. So I want to go through that really rapidly, and then I want to move on to a new topic, really. How do we integrate life in the soil with our monocrop agriculture, where we're growing a single species? Well, in permaculture, we know better than that. But still, even in permaculture, a lot of the times we're still growing a monoculture in a field for short, for 1 or 2 rotational stages. And really, how do we change that? How do we incorporate permaculture principles into our understanding of growing crops? How do we not have to rotate? How do we maintain the life in our soil when we don't have a crop in the system? So that's kind of where I want to go in the next, I think I have an hour and a half. So as you guys know from this morning, I could probably talk on for about the next 3 months on this topic. And so I will be summarizing a lot of the information. I can't go into the detail that I would like to go with you folks. So please, love to come back, or have some of you come to some of the other classes where 75 minutes? Yeah, hour and a half. So a little less. I have an hour and 15. Whoops. Well, okay, I'll talk even faster. So I'd love to have you come to some of the longer talks where we really get into the gory details of why the biology works. We get down to the molecular level, because really, if you want to understand what's going on and how to predict that something's going to happen or not, we really have to get down to that level. Well, for many of you, that's like, oh, I can't deal with molecular. Oh, that's way too detailed. So today's talk is going to be perfect. So when we're looking at the soil food web, we want to make sure we have the organisms in the soil that will set up the habitat, set up the conditions in your soil so it's inappropriate. It's not possible for the diseases, for the insect pests, for the problem organisms, for the weeds. They won't grow. They won't germinate. If we make the habitat inappropriate for these organisms, they won't be problems. But how do you do that? We have to get that soil aerobic. We must build structure in that soil. We have to have, voice is running out a little bit, so I move the mic closer. We want to make sure that we build structure in the soil, and who is it that builds structure? So if we're looking at this last part right here, who is it that builds soil structure? Well, bacteria are necessary. So kind of think of this as like a 12-step program. First step is to make sure that we're growing bacteria in the soil, because bacteria make the glues that bind and hold sands, silts, clays, organic matter, rocks, pebbles. They hold those things to your root system. They make the microaggregates in the soil. Bacteria make a massive amount of glue. And when we start looking at soils and we're trying to understand what's going on, microaggregates, the smallest structure in our soil has to be built by bacteria. It's kind of like thinking about a brick house. You can't build a house out of sand and straw. You've got to glue them together somehow. You have to bring them together into the smallest building block for a brick house, which of course is a brick. So that smallest unit of structure in a brick house, that's what a microaggregate is. So it's the bacteria that provide the glue to hold that brick together. Well, you're not making a brick house with bricks alone. You've got to have something to glue those bricks together and start building the walls. You have to leave holes for the windows, for the doors. You've got to put a ceiling, you've got to put a roof on that house, you've got to have floors. Who is it that's going to build those structures for you in the brick house structure in your soil? Those are going to be the fungi. The fungi build our strands, their filaments, and they're going to take those microaggregates and pull together a whole bunch of those. So we're starting to build the walls, we're building the doors, the ceilings, the floors of the structure in our soil. And as we start to build these structures, you can see where we're pulling things together, and now we have space for oxygen and water and our roots to move through that and grow through that soil. We've got to have oxygen so the beneficial organisms grow and the bad guys don't. The diseases, the pathogens, the pests require reduced oxygen conditions in order to outcompete the good guys. So maintaining these air passageways, the hallways, is a critical part of what we've got to do in soil. Now, when we think about the difference between soil and dirt, what is soil? What's dirt? So when you think about defining the four-letter D word in soil, dirt, we are looking at only sand, silt, and clay, the mineral fractions of the soil. You've all seen that triangle that describes whether you've got a sand, a silt, a clay. You're dealing with the clay loam or a sandy loam or all those descriptions of the soil. And really, we're not talking about soil at all when we're talking about that triangle. We are talking about dirt because those are the only factors that are in that triangle. And that's dirt, folks. Can we change a sand into a loam? Well, you could change the size fractions, but time and time again, we've gone out into agricultural fields where we've started with pure clay or we've started with pure sand. And we bring the soil scientists out and we ask them to take samples and define what kind of soil this is that we're working with. And it's a pure clay or it's a pure sand. And we just kind of go, okay, pure sand, pure clay, whatever. Great, it's documented. And then we ask, can we grow a crop in here? And most of the time, the agronomist is going to say, no, you're not going to be very successful. You don't have the nutrients. You don't have the ability to grow plants. You're going to have to load on the inorganic fertilizers. You're going to have to use pesticides, and they give us the whole 9 yards of what we got to spend to be able to grow a crop here. And we just happily nod at them. And we go out and we put on organic matter, put on compost. We have a lot of residues. We get some mulch. We put these materials on, allow the microorganisms to start building. Our microaggregates, our macroaggregates. We get the higher level creatures, the protozoa, the nematodes, the microarthropods, the spiders, the voles, the shrews, the mice, all of those things that build the bigger spaces, the pore spaces that when they're filled, we will retain and hold water within that soil. We've got to build that organic matter. Organic matter holds 10 times its weight in water. So let's hold and keep all of the water. Every time you have dewfall, we want all of that water held and kept within your soil. So we want to build all that structure. And we spend the summer growing crops, getting all of this going. And then we invite the soil scientists to come back at the end of the summer and have them come out and we say, okay, here's our sand, pure sand. And he goes, oh no, that's not sand. That's a loam. It behaves like a loam. And so we kind of giggle, and we let him take soil samples and take them back to his lab. And what comes back is, yes, this is a pure sand. But the organic matter content in that sand has built the structure. We've got the air passageways. We have the quality, the texture of a loam, because that's what biology does for you. I can take a clay and turn it into something that behaves exactly like a loam. We can take a sand and turn it into something that behaves exactly like a loam soil. And yet there's not a drop of clay. There may be no silt in there. There may be no sand. We don't have to have those mineral fractions. Because if we've got organic matter with good structure and the biology, that's what we need to grow plants. So can we do that with you folks? Any place we have these kinds of problems? Well, absolutely. And then we've got air passageways. So we have those organisms that don't allow the problem organisms, the diseases, the pests, can't grow. We've got the bacteria and the fungi that retain nutrients. We have to have the higher level predators that will cycle those nutrients into plant available forms. And quite often people just don't understand this whole nutrient cycling system. It's never been explained to you in the course of your educational experience. No one has explained to you how this works. So let me very briefly go into it. When we're looking at energy coming from the sun, carbon dioxide in the atmosphere, the photosynthetic organisms are going to build that organic carbon, those chains of sugar where we're storing sunlight energy between the different carbon molecules in that carbon chain. We're making sugars. Your plant makes sugars, but your plant can't grow on sugar alone. So where's your plant going to get all the other nutrients that it requires? From the soil. So those sugars have to travel down into that root system, and those sugars must pick up all those other nutrients from that soil. How does that happen? How does it work? How are those nutrients made available into your plant? So when the agronomist talks about this, simple diffusion pulls those nutrients into that root system. Okay, fine, but how did those nutrients become available in your soil? What is it that is constantly, every second of every day, replenishing that soluble, available form of nitrogen or phosphorus or magnesium or calcium or sodium or potassium or iron or zinc or cobalt or all 42 essential nutrients that are required by a growing plant? Could it be more than 42 essential nutrients? Probably, but okay. We got data for 42 essential nutrients for your plant to be able to grow. So how is it those are made available around the root system of your plant? Are you putting in those in there as a fertilizer? Because those just leach out. If we're putting in organic fertilizers into soil, we already know that 80% of an inorganic fertilizer is going to wash out of the soil, and those ions are going to end up in your drinking water. And now you're going to have to pay money to clean up those nutrients out of that drinking water. Let's keep them in the soil instead. How do you do that? We got to have the biology to retain and hold. Which organisms retain and hold those nutrients in your soil? Well, that's your bacteria and your fungi. Bacteria will grab different things. Fungi will grab different things. But on the other hand, what grows bacteria, what causes fungi to grow in your soil? They've got to have organic matter. Where's most of the food coming from to grow most of the bacteria and fungi in your soil? It's from the plant. So the plant is putting out exudates. And some of the exudates are specific for growing certain species of bacteria. Some of those exudates are specific for the fungi. Your plant is going to put out this kind of exudate to grow this species in order to solubilize iron out of the not plant available forms of iron in your soil. Or if what your plant requires is some more cadmium, plant's going to put out the exudate to grow those fungi that make the enzymes to pull those nutrients out of the soil. So, don't you love those scientific terms? What the heck is an exudate? Does everybody know what an exudate is? No? A bunch of no's? Okay, so what is an exudate? Gotta love scientists. We like to have our own special secret language, so you guys can't understand us when we talk. So I remain an expert, so you must come and pay me to come out and help you. Okay, here's a soil science secret. What's an exudate? Well, an exudate is basically the sugars that are made through photosynthesis that your plant is pumping down into that root system. Most of the exudates are going to be dumped out into the soil, those sugars, those proteins, those carbohydrates. So an exudate is mostly sugar, a little bit of protein, a little bit of carbohydrate. So if I send you into your kitchen and I ask you to make me a recipe of mostly sugar, a little bit of protein, a little bit of carbohydrate, what am I asking you to make? Yeah, cakes and cookies. So what's sugar in your kitchen? White sugar, brown sugar, honey, molasses, all those wonderful sugary things. So put some of that in your bowl. What's protein in your kitchen? Some eggs, milk. So put those into that recipe. What's carbohydrate in your kitchen? Flour. Yep, so let's put some of that in. What is this a recipe for? Cakes and cookies. So think of all the different kinds of cakes, all the different kinds of cookies that could be released from these root systems. And each specific kind of cake or cookie is going to grow the specific bacteria or fungi that plant needs to wake up, needs to get it growing to make the enzymes to pull the nutrients out of the sand, out of the silt, out of the clay, out of your rocks, out of your pebbles, out of your organic matter. And now we're going to retain those nutrients in that bacterial and fungal biomass while they are happily making structure for your plant to grow deeper and deeper into the soil, building that structure. Isn't it amazing how nature does more than one task with everything that she does? It's a little bit mind-boggling sometimes, learning all of this and connecting it all together. Okay, but great. We just tied up all our nutrients in the fungi and the bacteria. Any soluble nutrient sucked into the bacteria and fungi. Think about that root system. We've got a massive wall, a castle wall around that root, a bacteria and fungi growing and sucking up all the nutrients. So is it true that by simple diffusion of your nitrate from out here, that nitrate's going to get pulled towards the root system and move into the root? Not happening, is it? Because what's between your root and that source of any ion, any nutrient, soluble nutrient moving towards the root? What's between your plant root and that nutrient that's going to suck up and take that nutrient away? Now it's tied up in the bacteria and fungi. So you see, if you have soil that only has bacteria and fungi, you are in trouble. You got dead plants. So what else do we have to have? How has Mother Nature solved this problem? She doesn't want any of our nutrients washing out of the soil. So lots of bacteria and fungi. But now, how do you make those nutrients available back to the plant right there in the root zone? Not out here in the rest of the soil. We don't have roots growing. We don't need this nutrient cycling happening out here. We want it happening only around that root system. So, protozoa. Nematodes, the good guy nematodes, the good guy microarthropods, anytime they eat any of their prey, so anytime these predators eat their prey items, they are going to release nutrients in a plant-available form. And of course, where is this going on? Where's the most bacteria and fungi in your soil? Right around the roots. So guess where the predators are attracted to? Right there around your roots. So they eat a bacterium, they eat the fungi, they release nutrients in a plant-available form. But please remember, it's not just nitrogen that's released. It's not just phosphorus that's released. It's all of the nutrients. Every single one of those 42 essential nutrients plus others are going to be released in a plant-available form. So we're going to release our nitrogen as ammonium. We're going to release our phosphorus as phosphate. We release sulfur as sulfate. What's the form of calcium that's going to be released when a bacterium is eaten by a protozoan or a bacterial-feeding nematode? When a fungus is eaten by a fungal-feeding nematode or a fungal-feeding microarthropod? What's the soluble, plant-available form of calcium? Anybody know? It's not calcite. That's a mineral element. Plants cannot take up calcite. Sulfate, that's SO4. I'm asking calcium. Ooh, nobody ever explained this. No one's ever gone through that with most people. So what is the soluble, plant-available form of calcium? Chloride is chlorine. Calcium. It's chelated calcium. Ooh, there's another good scientific term for you. What the heck do I mean when I say chelated? How many of you have heard that term, chelated? Yeah. But we don't know what it means. When we say chelated, what does that mean? Pre-digested? No. Stuck to a protein. Stuck to an amino acid. That's all chelation means. So if we're talking about chelated calcium, chelated zinc, chelated boron, chelated, that's the form that your plant takes it up. All those other micronutrients. So where do those proteins come from? Where do those amino acids come from? As the bacteria and fungi are eaten by the protozoa, the nematodes, and microarthropods, those amino acids are released, and bang, now we've got our carrier to allow those nutrients to easily be taken up. And where is most of that chelated calcium, potassium, sodium, iron, zinc, boride, iodine, all of those things? Where's the highest concentration of chelated nutrients? Found right there around the root zone. It is so amazing how nature has put these systems together. And yet we didn't know that these things were going on. When I started graduate school, the project I worked on was trying to figure out what it is. Why is it we've got bacteria in the soil? Why do we have fungi? What importance to plant growth do protozoa have? Nematodes, who needs them? Let's just nuke them all, get rid of them. Yeah, good luck growing a plant if you don't have the normal natural nutrient cycling going on in that soil. So hopefully you're starting to get that understanding of the importance of these organisms in your soil. So now you understand why we've got the fungi, why we've got the bacteria, and of course it's the good guy bacteria, aerobic. Because the anaerobic guys are the diseases, the problems. They're making horrible compounds that will kill your plant. We went through aerobic, anaerobic. What happens when you go anaerobic? You're going to produce the most plant toxic material that we know on this planet. Only occurs in anaerobic conditions. Somebody says to you, oh, it doesn't matter. Your tea can go anaerobic. What problem could an anaerobic tea give you? It'll only kill your plant. So probably not something that we want to do. You're going to lose your nitrogen, your phosphorus, your sulfur as gases if you go anaerobic. How are you going to grow plants? If you have no available nitrogen, no available soluble sulfur, and no available phosphorus. Good luck. Enjoy yourself. Because how can you do it? Well, you can see why we've ended up having to put inorganic fertilizers out. The biology in our soil was destroyed. We lost these organisms. Our soils got compacted. They went anaerobic. We were producing extremely toxic things in our soil. So we got to rebuild this structure. You know, we got to have the higher level predators in the system, because if you get too many of these guys, can you overeat and wipe out your bacteria and fungi? Oh yeah. And if you have no bacteria, no fungi, say goodbye to the structure in your soil. Your soil is going to collapse, compact, Go anaerobic. And now we got dead plants. Okay, so we have to have these guys to control those guys. Well, okay, can we get too many of them? Because if we get too many of them, they're going to eat all of those guys, and now there's nothing to eat these guys. And so all our nutrients are going to get stuck, held in the bacteria and fungi, and what happens to your plant? Yeah, dead plants. No nutrition. So do you need everybody in this food web? Absolutely. Who's at the top? Now, as you go all the way up to the animals, and you keep going to the top animal, well, at least we think we're the top animal on the planet, don't we? We are the top of the food chain. So whose responsibility is it to manage this whole system and manage it in a way that's going to be sustainable and stop killing people? That's us. So you can see where it is so critically important to understand permaculture and to understand how the biology in the soil works with all the other things we're doing in permaculture to build fertility, to reduce the amount of work that you have to do. Put your willing organic workers to work for you because they will do all your work for you. Our only job is to make sure all of that diversity has gone out into your soils, that everybody who needs to be there is present. So we've got to have a massive diversity because every different species of bacteria or fungus requires different conditions. If we're trying to grow good soil at 55 degrees, it's one community, about 1,000 species of bacteria, another 500 or 600 species of fungi. We've got to have 10 or 20 species of protozoa. We've got to have a couple species of nematodes. But what if you shift the temperature to 60 degrees? It's a whole different community of bacteria grown at 60 degrees as compared to 55. A whole different species of fungi at 60 as compared to 55. So what if we shift the temperature to 70? Okay, now we need a whole different community. What about 75 or 80 or 85 or 90? What temperature does your soil reach in the middle of the summer? And you better have the species of bacteria and fungi doing their jobs, the protozoa, the nematodes, the microarthropods. How many species of bacteria and fungi do you have to have in soil? If you want to keep nutrient cycling going every second of every day while your plant's growing, every time your plant puts that exudate out, expecting that the organisms will utilize it, make those enzymes to solubilize the nutrients straight out of the sand, silt, clay, organic matter, rocks, pebbles, turn it into bacteria and fungi, get eaten by a protozoa, nematode, microarthropod, and release those nutrients in a plant-available form. How many species of bacteria? Per teaspoon, 75,000 bacteria per teaspoon. The next teaspoon over, oh, it's a different set of bacteria and fungi, protozoa and nematodes. We have a different set of species of those organisms. How many organisms in a cornfield? How many species of bacteria in a healthy cornfield where we don't have to use pesticides, we don't have to use inorganic fertilizers, we don't have weeds, we don't have any problem organisms? Oh yeah, the plant grows itself. You don't have to do the work. How many species of bacteria? About 1 million. Per acre of agricultural land, we need 1 million species of bacteria. We need about 750,000 species of fungi. We got to have several thousand species of protozoa, nematodes. Do we know what the names of all those organisms are? In the world of microbiology, and my degree is in soil microbiology, the Bible of bacteriology only lists 5,000 species of bacteria. We can't grow them in the laboratory. We don't know what conditions are required to get those organisms to grow in lab conditions. But we can identify them based on DNA analysis. So 75,000 unique and different strands of DNA per teaspoon of soil. And so our job is to put that diversity back into the system. How are you going to do that? Can you afford to buy 75,000 different inoculant cultures of each one of those bacteria? We don't know how to grow them. Where do they grow? In the soil and in compost. Yay! So grow your own compost. Grow your own organisms. The most important thing in compost is the life in that compost. That's the real reason we make compost. And therefore, when we're making compost, it has to remain aerobic. If our compost goes anaerobic, we're growing exactly the wrong set of organisms. We're growing pathogens. We're growing pests. We're losing our nitrogen as ammonia. We're losing our phosphorus as phosphine gas. We're losing our sulfur as hydrogen sulfide. We're dropping the pH in that compost maybe down to as low as 2. And can you grow plants in a soil with a pH of 2? Nope, not happening. We're making alcohol in that compost. If it goes anaerobic, it's not compost, because we are making the most toxic things that will kill your plants. It's not compost anymore. What should we call that stuff? That pile of organic matter that's gone putrefied or stinky, smelly, black stuff. What should we call that? Roundup. Okay, that's pretty good. Should we call it stinky, smelly, black stuff? Putrefied organic matter? It's not compost. And we all have to understand that when we walk up to a compost pile and it's black in color. Topsoil? No, not even topsoil. Because in our topsoil, we want all these good organisms. We want humic acids, not charcoal, not the toxic residues left behind when ammonia or hydrogen sulfide are being produced. So we have to keep the composting process aerobic. When somebody says, oh, it's just fine if it goes anaerobic, how could they possibly say that? They clearly do not understand the damage that occurs when something goes anaerobic. So try to understand that. So I also want to talk a little bit about microscopes. I want to talk about succession. What is the right set of microorganisms for the different plants that you want to grow? So jumping ahead, I've kind of gone through all of this, so I'm going to zoom along microscope work. Like to introduce you all to the concept of buying your own microscope so you can do these assessments yourself, or find somebody in the neighborhood, in the area, that will learn how to do this microscope work, so you can take your sample over to them, walk in the door with a nice representative sample of whatever it is you want to know about. I'd like to know about my yogurt. I'd like to know about my milk. I'd like to know the sediments in my pond or my lake or my river or my stream. I would like to know the organisms in my compost or in my compost tea or my compost extract. Anything you want to ask about. What's the biology in? They can do on the microscope. The microscope that we use it costs about $350 with the camera. It takes about a day for me to train you how to use that microscope so you can identify these organisms and start figuring out what it is you have in your soil. So, isn't she cute? Isn't that just the sweetest little nematode you've ever seen in your life? Woo! Oh, it's the first nematode you've ever seen. So, how do we identify that this is a good nematode? Well, we look at the morphology. You know, when you look at that nematode, we're seeing a mouth. There is no spear. Fungal-feeding nematodes make spears, puncture the cell wall of the fungus, suck out the internal contents. Root-feeding nematodes, they have spears with big knobs. Pretty easy to see those big knobs. So as soon as you see that spear with the big knob, your heart kind of sinks because, uh-oh, somebody else is getting my plant material. Reducing yield for myself. Taking food away from my animals. So how do you get rid of root-feeding nematodes? You've got to get the aerobic organisms back into that soil. Root-feeding nematodes are present only because we've got anaerobic reduced oxygen happening in that soil, and that allows the bad guy nematodes to beat the good guy nematodes. So looking at their morphology, what does it look like? Turn around and look at the organism next to you in the next chair. How do you know that's a human being? It's got a head, legs, arms. Yeah, I think that's safe, isn't it? We call that a human being. Now, is that a good guy human being or a bad guy human being? How do we know this is a good guy? Well, she's here with us, right? She's at this meeting, so must be a good guy. The habitat, here's the environment. This is a wonderfully aerobic environment. Positive, we're going to move forward. So, how do you tell? You got to look at the habitat that it's in. Is it anaerobic, aerobic? Is the morphology that of a good guy or not? So, look at this picture. Are these good guys or not? Uh-oh. This guy right there, that's a ciliate. It's a kind of protozoan, but it's a protozoan that only grows as oxygen concentration starts to drop into the anaerobic conditions. Reduced oxygen allows these ciliates to win in competition with everything else. How can I tell if it's a ciliate? See those little legs on it, the little hairs? Those are called, oh surprise, cilia, therefore ciliate. But when you see these guys, oh no, you just want to cry because it's telling you got some anaerobic microcytes in here. Now, nice aggregate right there, our little bacteria. We got some rod-shaped bacillus bacteria. We got lots of little cocci, little round guys. So we've got a nice aggregate. We've got more aggregate, more. Look at the color in there. That honey-colored material, tan, that's fulvic acid. So we've got organisms that are condensing that organic material down, putting savings accounts away. We're storing nutrients for hundreds of years as fulvic acids. When we look at the dark brown colored material, that's all humic acid, made by, condensed down by all this wonderful life in that soil. Mostly fungi make humic acids. Mostly fungi eat humic acid. So, putting it away, taking it out. Constantly maintain that really good fungal community by having these really good humic acids in the system. So once upon a time, this was a good soil. Is it still a good soil? Because that cilius tells us, no, something bad's going on. Second thing is a problem. See this root system, or see that root right there? Roots should be uniform diameter all the way along that section of root. But notice we've got a big divot. We've got a disease-causing fungus attacking and consuming that root system. Another piece of evidence that says it's gone anaerobic because these disease-causing fungi will only be able to attack and consume your roots when we fall into reduced oxygen conditions. Our plant's going to suffer, perhaps even die. So we don't want to see this kind of damage. When you see it, you should start to fix the problem with the biology as fast as possible. Get the organisms back in here to rebuild structure. Get that oxygen, get the water moving through the system, held in the soil so your plants can always find that water through the driest of conditions. We have soils in Australia, for example, where we have had drought for the last 38 years. We have been extremely under any normal rainfall, and yet we grow plants, keep them alive, normal productions, even when all the surrounding or conventional systems make no yields. Done that over and over again. So if we get the structure back in there, don't have to worry about water, but we can't allow compaction or anaerobic conditions to occur. Good guy fungi. So nice strands. Fungi are uniform diameter all the way along. They're wide diameter, so we're looking at about 4 micrometers here. There's another fungal How many of you saw this fungal hypha right here? The guys who are in the session this morning. A little bit wider diameter. We have another fungus right over there, about 3 micrometers. Colored fungi, always typically good guys. Clear, colorless, narrow fungi, uh-oh, there's a bad guy. We have a disease-causing fungus in there. Am I concerned about the fact that in this soil I've got a disease-causing fungus? No, not at all, because I've got all these other aerobic, beneficial organisms that are building structure for me and keeping this soil aerobic. So using the microscope to help you understand what's been going on in that soil. So over the years, as we've worked on lots and lots of different systems, the first time we began to understand what is the ratio of fungi to bacteria. So when we're looking at fungi to bacteria, what is that ratio as we go through succession? This work started when I was at Colorado State University working on my PhD and then stayed to work on a postdoc for a couple years. We did an assessment of irrigated wheat fields in eastern Oregon, or eastern Colorado. We looked at dryland wheat production out near the shortgrass prairie, the Central Plains Experimental Range. Looked at the shortgrass prairie, what's the biology there. Looked at the tallgrass prairie, looking at high mountain meadows, and then finally old growth. So it's a productivity gradient going through time. And we started to show that very early in succession, where we're mostly having weeds in that system, the fungal to bacterial biomass ratio is about 10 times more fungal than bacterial. As the weeds, and that's what we're selecting for in that kind of system, as the weeds grow, they are contributing a little bit of fungal food to that soil, mostly bacterial, a little bit of fungal. Disturbance requiring weedy species, not plants out of place. That's a stupid definition of weeds. So true ecological definition of weeds require high nitrate levels, strictly bacterial, not good structure in the soil. The weeds are going to do just fine when you've got compaction. And so when that fungal to bacterial ratio is very bacterial, setting the stage for those weeds to grow. But weeds are, eventually they will, put enough fungal food back into the system to shift that ratio of fungi to bacteria into something that's going to promote the growth of early successional grasses. So Bermuda, bromus, cheatgrass, oh, lovely grasses. Brassicas, so the non-mycorrhizal plant species come next in succession. But they are going to put a little bit more fungal food. They got a little more cellulose, a little more lignin in that plant material. Structurally, a little bit more stiff, strong. And so because we're putting more fungal food in, or nature has put more fungal food in, that fungal component is going to increase. And so that fungal to bacterial biomass ratio shifts, and now all of a sudden the plant species shift as well. We're going to be growing tomatoes, potatoes. We're going to be growing carrots. Our middle successional grass species, ryegrass, such as we find in your lawns. They are putting more fungal foods into the soil than bacterial. And so sooner or later, we're going to shift and we're going to have an equal biomass of fungi to bacteria, and we set the stage to grow all of our row crops, all of our corn, beans, barley, wheat, rye, all of those things, late successional grass species. The most beneficial when we're dealing with pastures, we're growing our blue and black grama. We're growing big and little blue stem. We're growing the best species of grasses to feed your animals, keep them alive. As we continue to put more fungal than bacterial foods into these systems, we shift over to shrubs, vines, bushes. A lot more fungal food than bacterial. And so we're going to continue to shift into the truly fungal-dominated systems till we get to the old-growth climax communities with extremely wide fungal to bacterial biomass ratios, as much as 100 times more fungal than bacterial to maybe 10,000 times more fungal than bacterial. Now, a lot of people will take that as, oh, well, it's just strictly fungal dominated. That's all that's in there are fungi. No, 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 no. That is not what I said. We still have the same amount of bacteria. So pretty much all the way through succession, we need the same amount of bacteria in the soil. What changes are the fungi. So, how do you figure out what your plant requires? If you wanted to grow strawberries, what is the proper fungal to bacterial biomass ratio for your strawberries? Figure out where strawberries are on the successional system. In nature, in the natural system, where do you find strawberries growing out in the real world? They're understory plants in forests. So they grow under deciduous trees. They grow in conifer forests. So what's the proper fungal to bacterial biomass ratio to grow strawberries? Yeah, there we go. Now think about here in California. What do we do to the soils before we put our strawberries in? We methyl bromide them. We go out there and nuke the biology. So we're trying to grow strawberries in what kind of soil? Yeah. So no wonder we got weeds. No wonder we've got diseases. No wonder you can't grow strawberries for more than 3 to 4 months. And they're wiped out by diseases and pests. And what do those strawberries taste like? Wood. That's not strawberry. What do you call that stuff? Filler? Carbon? Very good. So no wonder we're not successful. So start thinking about where your plant is on the successional sequence. When you're going, I just don't know, I can't figure it out, then you email me. And I send back the information. In Dave Jackie's book, there's a table of what the ratio of fungi to bacteria need to be in different kinds of crops. So we did about 350 different plant species, and that table is in that book. Now, when we look at those numbers, so I'm giving you some examples of, 10 micrograms of bacteria, no fungi. Okay, bare soil. We're not growing much of anything. Well, then weeds come in. And so in a really weedy system, 100 micrograms of bacteria, 10 micrograms of fungi. Keep going. Now we're growing brassicas. We're growing kale and cold crops. We're growing non-mycorrhizal plants. There you go. There's our ratio of fungi to bacteria. Now we're growing vegetables, row crops. We're growing the things that require equal biomass of fungi to bacteria. The more we can get that bacterial and fungal biomass ratio up, the more productive the system's going to be. If we got 300 to 300 fungi to bacteria, that's minimum. We're going to just barely make a decent crop. If we can get 400 to 400, we're going to double or triple yield. If you can get 600 to 600, we may see increases of 10-fold, 150% more crop production as compared to conventional systems as we continue to build the biology. That means we're building more nutrient cycling. Our plant's getting fed faster. It's going to grow faster. It's going to be more healthy. Working with Great Grow in Pennsylvania this last year. It's basically a compost material that you add into your soil with really good sets of bacteria, fungi, protozoa, and nematodes. Mycorrhizal fungi going into these soils. Our corn in the chemical system had 2 layers of corn ears, 2 ears of corn on the bottom, 1 ear of corn on the second, and that's a pretty good yield of corn, most people would say. But in the biological system, we had 5 rungs of corn ears, and there were 2 ears on each of those levels. So we increased yields. Now, is that going to happen year after year after year? Best of our knowledge, yes. If we can maintain this biology, but you see what's important? It's maintaining the biology. We got to have the bacteria and fungi. We have to have the protozoa and nematodes. We got to have the mycorrhizal fungi in those systems. As we keep going in succession and we start growing grapevines or blueberry bushes or shrubs of any kind, we've got to get this actually bacterial, more bacterial. But notice the bacteria are hanging out about the same level all through here now. What's changing is a fungal component. When we get into our old growth forests, 700 micrograms of bacteria, 7,000 micrograms of fungi. In the most productive, healthiest old growth forests, this number should actually be 70,000. So when we're in the Olympic Peninsula, totally fungal dominated, extremely rapid growth of our trees, 70,000 micrograms of fungal biomass per gram of soil. That means 75% of the weight of a gram of that soil, a teaspoon of that soil, 75% of the weight of that is fungus. Do not tell me that microorganisms are only 5% of the mass of a gram of soil or a pound of soil. Well, okay, maybe if you're in a sick, unhealthy, unhappy, chemical, agricultural field, maybe microorganisms are only 5%. But when we get healthy, life is 75% of the weight of a gram of soil, a teaspoon of soil. As we go through this shift from very bacterial to very fungal, the chemistry in that system changes completely. When we're growing bacteria, the glue layer that those bacteria make are alkaline. Aerobic bacteria make a massive amount of alkaline glues. So the pH in a bacterial-dominated soil is going to be what? It's going to be alkaline. When fungi grow, they release organic acids. Fungi don't make those glue layers. So as we move along this system, as fungi grow, they release organic acids, and they're going to buffer the pH of your soil between 5.5 and 7. So who's in control of the pH of your soil? pH is a consequence of whether you're growing fungi or whether you're growing more bacteria. And if we're in a bacterial-dominated system, the NH4 released by bacteria and fungi being eaten by protozoa, nematodes, and microarthropods, the NH4 that's released is going to stay as NH4 when we're in a fungal-dominated system, when we're slightly acidic. As we go over and we become an alkaline material, when the bacterial become predominant and our pH of our soil is now above 7, now our nitrifying bacteria will produce the enzymes that will rip the hydrogen off that NH4 molecule and replace it with oxygen. So if we're in a strictly bacterial-dominated soil, all we've got basically are bacteria. Nitrate's going to be the only form of nitrogen, and that grows weeds. As we shift and we get balanced, what our row crops, what our veggies require is a balance of nitrate and ammonium. Our shrubs need more ammonium than nitrate. Our old-growth trees require almost strictly NH4. So chemistry in soil is a consequence of what the biology is doing. So I like this picture. This is work that we were doing in onion fields, because a lot of people don't believe that we can really get rid of weeds when we use the biology. So this is a conventional onion field, working with this onion grower. He'd been conventional for about 50 years and decided that he wanted to test out this current rage of compost, compost tea, compost extract. I do like to point out that most of the work that we're doing with compost, compost tea, compost extracts, we're working with a lot more people overseas than we are in the United States. Because really, you have to think about the U.S. as being pesticide central. The chemical companies, the fertilizer companies, all that big agrochemical conglomerate have really got things in the U.S. pretty well tied up. As we go overseas, A lot more interest and a lot more willingness to work on these systems overseas. So Tasmania, which is where this work is being done, the government is paying people to put out compost, compost tea, compost extract, and show that it works or it doesn't work. So he decided that he wanted to do that work. So Ian said, 150-acre onion field, I'm going to put on the conventional. So this field has been treated twice with Roundup. The onions were planted on the same day, a few weeks previous to this picture. And you can see how well the Roundup is dealing with the weeds, which is sort of like not. Can you see the onions? Kind of hard, isn't it? You know, can you see the onions over there? Whoa. Now, when you're straight on a row, okay, you can see them. But those onions aren't very tall, aren't very healthy, aren't very happy. And the weeds, wouldn't you say the weeds are winning? Yeah. The field right next door. We had, by this point, the onions had been planted on the same day here as in the field next door. But next door, we've applied 2 applications of a really good compost tea. Can you see any weeds? And so we've really dealt with the weed problem. The onions are taller, healthier, happier. When we look at the onion seedlings in the chemical field, the seedling's only maybe 3 to 4 inches tall. The root system's only going down 2 to 3 inches, and it's already diseased. We're already picking up pink rot right there on that root system. Yields were nowhere close in this onion field as compared to next door. When we're looking at the onion seedlings on exactly the same day, look at the structure in that soil. Oxygen, water, roots are going down deeper and deeper and deeper into that soil. Healthier and healthier plants growing better and better. The seedling is 6 inches tall. The root system on that plant is already at 1.5 feet. So these kinds of systems, we can deal with the weeds. We can move things along in succession rapidly if we understand what we're doing. But when you go in and disturb your soil, and we disturb soil through tillage, we may disturb it through pesticide applications, through inorganic fertilizers. Even in the organic world, we're allowed to use pesticides. Copper sulfate, it's a pesticide, it kills things. So we're allowed in that organic world to still use some pretty toxic things that kill the biology. So which way are you going in succession when you start destroying the life in your soil? We go backwards. So if you go out and disturb your soil by running a herd of cattle over it at the wrong time of year, compacting, we're going backwards, and the productivity of our system is harmed. Nature sends disturbances. What's a fire? How far backwards in succession are you going when a fire happens? So think about Yellowstone National Park about 25 years ago. How much damage happened? Well, there were areas in that fire where the fire just went really quick across the surface of the soil. Did not kill the trees, just kind of, toasted them up a little bit on the bottom. So sometimes it doesn't do anything. Leaves a little bit of fertilizer, ash, and the trees grew better the next year. Now in other places, the fire burned the trees, it burned the organic matter on the top of the soil. So we came back to here. It did not actually burn the soil or any of the organisms in the soil. And so the next year, we had some really beautiful grasslands. They came up. The seed had been waiting for those stupid trees to be gone. So up came the grasses. The buffalo herds did better. The elk herds did better. They had more range. The herds increased. But some places, when that old-growth forest burned, the temperature got so hot for so long, it actually burned down into the soil to the point that the silica in the soil melted. And the surface of the soil turned into glass. And when I went back, I was invited back by the U.S. Forest Service, Park Service, to come back and look at those areas. So about 3 years ago, I came back, took a look, and there were still areas that were covered with glass. Just starting to get some plant material coming up. So when we go backwards in succession, How long is it going to take for Mother Nature to start moving that successional process forward? And then another disturbance comes along, and then she starts rebuilding, and another disturbance, and she starts building, and then another disturbance, and she starts rebuilding. What's the yin and yang of what's gone on your property? Where are you? You got to have that microscope so you know where you are. What's home? What do you have to fix? So I'm going to jump through this, these a couple examples. I do want to point out, Mother Nature abhors bare soil. If there's bare soil, what is Mother Nature going to put in there? So let's not allow her to do that. Let's not have bare soil. If we tilled it up, and now we've got soil that doesn't have any life, doesn't have any plant material in there, let's us choose the seeds. Let's us come back along and put in some low-growing understory plant species. Let's us choose. Let's us make that jump start. So instead of letting Mother Nature decide, something happens, now we're back here. Let's not let Mother Nature do this. Let's us come back in with what stage of succession do you want? Let's put in some understory plant species that don't grow more than 1 to 3 inches tall. That those root systems go way deep down into the soil. How far down into the soil do root systems go? Well, let's start picking those plants who put their root systems down deep. This is ryegrass. It was planted as seed in July of 2002. In November of 2002, 3.5 months later, we took a cylinder, pounded it down into the soil, and then lifted that up. Took a saw and took the PVC pipe off, used a hose and washed the soil off out of that root system. And there's Hendrika Schraven, who is a 6.5-foot tall person, holding that grass plant at his waist. The root system, in 3.5 months, has grown 4.5 feet. Is this as far down as your ryegrass will go? This is only 3 months old. Keep letting that plant build soil structure. Get the bacteria and fungi building the structure so that root system can keep growing. What's the maximum depth that we've ever found ryegrass? How far down can it grow? 25 feet. If you've got a really healthy plant, it should be down there at 25 feet. So let's us start choosing what plants are going to go deep down into the soil so that we have this food web and we're maintaining it every second of every day. So we want to completely cover the soil surface. We want plants that will grow out in a prostrate fashion. We want to choose things that have deep roots that will go down as far as we possibly can. So what are those plants that have all these characteristics? If we have non-mycorrhizal species in this part of our agricultural fields, our understory plants should probably be non-mycorrhizal. But if we want to grow mycorrhizal plants, we want to choose those species that will have the mycorrhizal associations on those root systems. In the world of chemical conventional agriculture, they have done absolutely the wrong thing in choosing cover crops. Totally idiotic and stupid. Let's, be careful in our terminology here. They've chosen for the wrong thing. They keep trying to grow plants that have massive above-ground biomass. This is biomass in the wrong place. When you cut this stuff, how are you going to get all that massive amount of above-ground plant material into the soil and turn that material into soil? How are you going to do it? You cut that plant material and it lays on the surface of the soil. If you've got a nice, thick, good growth, all that's going to happen is this is going to go anaerobic. And you're going to be killing whatever you try to plant in there. If you till this material into the soil, think of the oxygen demand of the bacteria and the fungi growing on that green plant material, or even if you let it brown down a little bit. Think of the oxygen demand, and you're going to go anaerobic. You till this material in the soil, and so your plow is pushing against the soil here. You fluffed and you got all that good organic matter into your soil. Where is it going to go anaerobic? You just compacted your soil down here. Lots of bacterial, fungal growth, goes anaerobic. Have a hard time growing your plants. It's the wrong attitude. Annual cover crops. You've got to go out and buy seed every year. Wrong approach. So in the permaculture world, we want perennial plants. In all of the surface of our soil, we want perennial seed that we're putting in there. Completely cover the soil surface. We want to have no real short foliage, 1 to 2 inches, 3 inches perhaps, no taller. Deep roots going down to constantly provide the exudates year-round, especially in this environment, year-round. So you don't have to feed your organisms anymore. How many of you enjoy putting out wood chips every place on your property? How many of you like all the time having to be out there, put out mulch, put out materials as a weed barrier or to feed your organisms? So let's stop doing that. I am the world's laziest gardener. Don't make me work. I want them to work. And so grow these perennial, permanent cover plants. So if you think about that growing season, kind of growing, perennial. The work that we've done with Gabe Brown, with Jay Fuhrer up in North and South Dakota, they put in a mixture of 25 different species of perennial, short-growing, understory plants. In the first year, they saw three or four species of those perennial plants came up in the first year and did a really good job of covering the surface. They decreased water use because you build structure. You're providing the food to feed all those organisms. They didn't have to use fertilizers. They didn't have any problems with pests. No problems with diseases. They didn't have rusts because we've got the biology to make sure that those plants had all the nutrients that they required and they could resist all of those problems. The next year, after a really wonderful, frigid, cold winter period, a different set of plant species came up in the second year. Why did something different come up? Well, it's a different weather conditions. One year was hot and dry. The next year was cool and rainy. The next year was rainy and hot. The next year was cool and dry. And the next year, have you ever experienced exactly the same weather in any year that you've been alive as compared to a previous year? It's always different, isn't it? So who's going to come up? Which of these species are going to do really well in any year? Hedge your bets. Put in a mix. So every year, something's going to be protecting your soil surface. And then think about the fact that you don't have to worry about when you're going to get into your field and plant, because you've got these understory plants that will always be the surface for you to drive over. When you're going to go in with a transplant, all you do is go in and dig your hole, drop your transplant in, and you've knocked back that understory. So now it's going to take a while for them to come back in, grow back in, during which time your tomato is going to get tall enough in shade. Your corn's going to get tall enough in shade. If you're putting in soybean, well, you do a strip, and that's where your seeds are going. And you make sure the strip is wide enough so that your seed germinates and grows. And what we find is if we put our seeds in with a really good active food web, where that mycorrhizal colonization is already functioning, already there, the day that seed's germinating, the root system of that plant is getting colonized by the mycorrhizal fungi, and now these understory plants are contributing nutrients to whatever plant you're growing. The seed germinates, starts to grow, you reach first true leaf stage in half the time. Get a jump on everybody else. The soil's nice and warm. Rain, when you start thinking about rain, it's one of the most compacting things that can happen on your bare soil. Drop of rain falling from the heaven, when it smacks on your soil surface, it's causing compaction. In tropical systems, the most compacting thing that is present in tropical systems is rainfall. You must protect your soil surface from that compaction. What can we put out there that's going to intercept rainfall before it hits the surface of your soil? Understory plants. Let it smack on the surface of your understory plant species. The drops roll off. They get into the soil because we've got good structure maintained by the exudates coming out from these understory plant species. The water moves in and stays. Oh no, if you've got plants growing on the surface of your soil, it's going to take water away from your crop. No. Where's the data come from that suggests that if you have plants growing in between your rows of crops, that's going to take water away from your crop? Who did that research? It was an herbicide company. And why do you suppose they chose the weeds that they did? Because yes, weeds, they are not efficient on water. Think about a weed. Germinates, it grows, it's going to suck nutrients out of your soil, suck the water out, because what's a weed doing? It's immediately going to go to seed. Does it care about how much water is left behind in the soil? Not one iota. So billions of seeds, and off they go to find another place. We have these understored perennial plants. Are they careful with their water? Yeah, because they have to stay alive. They need that water. So they protect the soil surface from evaporation. We reduce water use in those systems, again, by 70%. How many of you would like to lower your watering bill by 70%? So let's do it. We have a couple of different ways. How are we going to manage to reduce and make everything permaculture? What are the right understory plants to match up with your overstory crops? And think about the fact that you only have to buy these seeds once. And then they keep growing. Different sets, different mixtures come up every year, but you never again have to buy seeds. Annual cover crops, you've got to buy seeds every year. Who's making money? Is it you? Or is it the seed companies? So we've got to rearrange our thinking here to support this soil food web all the time. So I've gone over this. What are examples of these plant species? So here's the list. Is this the right set of organism, of plant species for your particular place? If you're on the north-facing slope or the south-facing slope, which are the right ones? See, that's what we got to learn. So if you want to get this list, Carol Ann Rollins out at the booth has these lists of understory species, and just go grab one of those from her. And as you try these out, I'm suggesting to you that you do it in small areas. Let's not go gangbusters. Try it in this, 100-foot by 100-foot area. See what happens. Which ones are the best species for your system, for your crop, for your soil, for your water, for your hydrological situation? And then I would like to have all that information come back to me so we can collate all of that. Put this up on the website and say, okay, in this part of California, these things worked really well, and here's a bunch of pictures. And in this part of California, it was more, these were better. Are there more species in this that might work? Probably. Let's put our minds together. Because how many years of experience do we have sitting in this room? Mind-boggling. Let's work together on this and really make all of our agricultural systems totally a mix of these plants, so we maintain and keep all of this life happy and living, and your root systems go as far down into the soil as they possibly can. They are never water limited. They are never nutrient limited. So let's work together on this, please. Thank you very much.